almost. You have to put in energy. Your anabolic reactions require energy input. Can't build something unless you're putting some energy into it. Alrighty. When I do transcription, I copy the DNA for one gene. I'm going to have to build the mRNA with nucleotides, and I'm going to need RNA polymerase enzymes to do the work. In translation, don't need my original DNA anymore, because now I'm going to work off the mRNA. So I need my mRNA, and in order to read my mRNA, I need ribosomes, that's rRNA, and I need tRNA to do the translating. And I need amino acids, because that's what they're translating it into, and I need enzymes. Still need RNA polymerase enzymes to build protein. So for transcription, you go from DNA to mRNA. But in translation, you need all three kinds of RNA. You need your mRNA that you just made, you need your rRNA and your tRNA to finish the job. Okie dokie. What kind of a molecule is DNA and RNA? Nucleic acid. Nucleic acid is the polymer, the big macromolecule. What are the monomers, the building blocks? Nucleotides. And nucleotides contain a sugar and a phosphate, and what's bonded to the sugar? The bases, right, the nitrogenous bases or the nucleotide bases or whatever it is you want to call them. A, C, T, and G. If it's RNA, U, right? RNA is single-stranded, whether it's mRNA, tRNA, or rRNA, they're all single-stranded. They're just in three different forms. mRNA is linear, rRNA is globbed up into a ribosome, and tRNA is in that funky turkey foot shape, the tRNA. Did I say that correctly? Okay? DNA and RNA are made up of the same things. They're both made up of nucleotides, but the nitrogenous bases are a little bit different. DNA has A, C, T, and G. RNA has U instead of T. How many codons are there? How many combinations of A, C, T,
64.
and U are there?
64.
64, good. How many amino acids does that code for?
20.
And what does that mean?
Redundancy.
The redundancy, right. Our genetic code is a triplet code, consists of combinations of 3 letters. There's 64 possibilities, but only 20 amino acids, so our genetic code is redundant, or degenerate is the official term. Redundant, good. So you would see the redundancy of the genetic code if you saw 2 codons that are very similar, coding for the same amino acid. They're similar, but they're different in which position, probably? The third. And what's that third position called?
The wobble.
Wobble. So that's the wobble effect. Good. And if I have a mutation where just that one letter is changed, what kind of a mutation is that called? Remember, a base substitution?
Silent.
Yep, a base substitution mutation means one letter is changed. 1 point. It's also called a point mutation. If it's in the wobble position and I get the wobble effect, that means it results in a silent mutation. So in a silent mutation, I have a different letter in the third position, the wobble position, but I get the same amino acid anyway. So my protein is unchanged, so my mutation is silent. What if the letter that was substituted with the wrong base, or I had a point mutation in the first letter, and I got a different amino acid? What kind of mutation is that? Missense, right. What if I had a change in the letter, and it resulted in no amino acid? Nonsense. Now, actually, those frame shift mutations result in the nonsense usually. Because what happens is it doesn't change the letter, it blocks the letter completely, shifting the reading frame of the gene. And that usually ruins an entire protein, so no protein results. So your frame shift mutations are the worst kind. because one of the letters was actually blocked and it shifted the reading frame of the gene. So it usually results in a nonsense mutation, no protein. All right. What is the word in our genetic code for one amino acid? What's that three-letter thing called? Codon. Good. So you know what a gene is? That's a segment of the genome or a segment of the DNA that codes for one protein or one trait. Genes are written in codons. Codons consist of nitrogenous base letters. Good. And one more time. Why does a cell make an RNA copy of the DNA? What's it going to do with it?
Make a protein.
Make a protein. Good. Where does transcription, making an RNA copy of the DNA, happen in a bacterial cell?
In plasma membrane.
At the nucleoid. Where are you going to make RNA? On the DNA. Where's the DNA in a bacterial cell? The nucleoid. So RNA at the nucleoid. In a eukaryotic cell, in the nucleus, and a prokaryotic cell at the nucleoid. Where does translation take place in a bacterial cell?
Cytoplasm.
Cytoplasm, good. On a ribosome, good. And let's see. Okay, on your transfer RNA molecules, one end you have the amino acid, and at the other end you have three letters that are going to complement the mRNA. On the mRNA, the three letters is called a codon, but on the tRNA, what is the complementary three letters called? Anticodon. Good. So in DNA, it's called a codon. In RNA, you still call it a codon, but it's got U's instead of T's now. Then in tRNA, it's got U's instead of T's, and we call it an anticodon. Okay. What is the name that we call the DNA molecule that Watson and Crick called it, describing its shape? Double helix, right. Why is it called a double helix? It has two strands of nucleotides. That's the double part. Helix, because they're twisted together. They're both helical twisted together. So it's two strands of nucleotides twisted together. And what holds them together in the middle? Hydrogen bonds. And what holds together the side rails? Covalent bonds. Good. Alrighty. Okay. Let's talk about those horizontal gene transfer mechanisms. Let's talk about conjugation first. In conjugation, F-positive cells contain a what? Plasmid. And how are they going to pass the plasmid to another cell? What else do they have? There's a gene for it on the plasmid? Pilus. Good. And he's F plus is like male. He's going to hook up with an F minus who's female. When he passes the plasmid to her, the copy of the plasmid to her, she becomes F plus. And for that reason, they call it an F plasmid. So on your test, you might see F plasmid. It's the same thing as a plasmid. It's just referring to the fact that it's got that F plus, F minus thing going on. So an F plasmid means you're transferring the F to it. If one cell is F minus and she receives a copy of the plasmid, she becomes F plus. Which makes her no longer a she, then she kind of becomes like a he. A lot of that going on these days. And apparently it's been going on for a long time, and now we're just finding out about it. Like every other person seems to be transgender. I don't know. Sometimes I wonder about those people you see at the MAC makeup counter, you know? Gee, that's a tall woman. They always get all the attention because they know they're going to buy a lot of makeup. You know? Like, I just want one eyeliner. Oh, we can't be bothered with it. And the guy needs a serious pancake makeup. Alrighty, let's see. In transduction, a piece of genetic material from a first host cell is transduced to a new host cell by boom? By a virus, good. So a bug or a virus would have to be involved. In transformation, it's a magical thing that happens, but it can only happen if the bacteria taking up the piece of DNA from the dead cell are what? Competent. So bacteria that can pick up the used DNA are called competent. They have to be competent.
It's interesting, they want to pick up DNA from a dead cell, it's dead.
I know.
That usually means it failed.
It's the recycling. See how advanced bacteria are? They've been recycling all this time and we're just starting to figure it out. They're smart bacteria.
I guess that's kind of like an organ transplant for.
Yes. All right, let's have a little flashback, because you will get some old questions. Some of them have to do with normal flora. You're going to have to be able to name some normal flora of the body. And also, remember, what do we use blood agar for?
Pathogenic bacteria.
Right? And why? What kind of enzymes do they have?
What'd you say?
What kind of enzymes do the bacteria have that we grow on blood agar? Why do we grow them on blood agar? Because they need to eat blood, right? What kind of enzymes do they need to eat blood? Hemolysins, right. You grow your hemolytic bacteria on your blood agar, so they have hemolysin enzymes. Actually, when we do chapter 14, the epidemiology chapter, I'll teach you about coagulase,